Hello, everyone. Thank you for joining me in this talk, which I hope will give you an overview of the next generation solutions that Akron is developing and providing to address the bottlenecks that the cell and gene therapy field is facing nowadays. Akron started back in 2006 as a premier provider of GMP cytokines and growth factors to enable the development of advanced therapies. And being testimony of the exponential growth of the industry and its needs towards commercialization, the company has embraced the challenge, adopting new products and systems that support industrial scale, fully GMP compliant and automated production. So along that road, Akron has always tried to be a pioneer in the industry, launching several products that have represented different inflection points in the field, like the virus-inactivated Chino-free human blood-derived products, or the first liquid GMP IL-2 in the market. Akron's acquisition by Arcline in 2019 enabled the acceleration of such growth to keep being in the front line, anticipating the needs of the industry, and proof of that are the multiple milestones that have been reached in almost only 2 years, like the electronic drug master files that we have filed with the FDA for IL-2, 7, 15, and 21, the new GMP plasmid facility that Akron has built, or the addition of our newest member of the family, the viral inactivated AV serum that I will present in a bit. And one of the milestones that we are most proud of is the first commercialized drug that has been supported by one of our electronic drug master files. As I mentioned, Akron started in the business of cytokines and growth factors production, and as the field has been refining and demanding top quality raw and ancillary materials, Akron has focused its attention on meeting these expectations, offering fully GMP, highly pure materials. As the industry keeps expanding, we realized how important it is to bring solutions that support the commercial scale and the regulatory compliance. And examples of that are our fully GMP large-scale manufacturing, using pharma-grade purification methods that result in cleaner and more pure cytokines. Also being the first with electronic DMFs covering IL-2, 7, 15, and 21, or the available cytokines in liquid-filled syringes. As we are aware that the field is moving towards functionally closed systems, we have leveraged our many years of expertise on the liquid formulation for IL-2 to also optimize and bring IL-7, 15, and 21 in liquid formats with 24 months of stability, which will be available in single-use weldable bags soon this year, similar to the one that you can see here in the image. Akron has, over the years, been focused on the immunotherapy field, building an exclusive catalog that has supported mainly T cell therapies, but as the field has been expanding and exploring other cell types, and this conference is a result of it, Akron has incorporated new cytokines and growth factors demanded by other therapies like NK or pluripotent stem cell therapies. And I think it is very relevant to this audience to know that we will soon bring IL-12 in a therapeutic grade. To demonstrate our commitment with the field, we have run several studies to show the equivalence between Akron's products and some of other ones available in the market. For example, this study was made in collaboration with Dana-Farber Cancer Institute. And what you can see here is the equivalence between Akron's IL-2 and Proleukin. Nowadays, one of the bottlenecks that the industry is facing, and that due to the current situation is more present than ever, is securing the ancillary material supply. So it is critical to have multiple validated suppliers in order to mitigate any disruption in the process development or manufacturing. And we must take into account that at the end of the day, it is the patient's lives that are at stake. So consistency and reproducibility of cytokine activity will strongly affect manufacturing costs, timelines, and overall quality of cellular therapies. And in order to have multiple validated suppliers, one has to make sure that the same product from different vendors has equivalent performance. This is especially difficult when comparing cytokines from different manufacturers due to two main reasons. So one is that some manufacturers have a huge batch-to-batch variability, and the other one is that today there is no standardized analytical procedure to determine the biological activity and a specific activity for the cytokines, resulting in inconsistent results when using the reported activities. So these two factors make very difficult to switch from one supplier to another, something that it is more needed every day to secure the process. So since at Akron we want to make things easier for our customers, we have invested some resources to find a solution. So first, as you can see here, we have consistent trend in the activity among different batches. And secondly, we have run several empirical studies having different manufacturers site by site and standards, measuring the potency with a parallel line approach, which is the approach currently used by the World Health Organization for the international standards. So these studies allow us to normalize and find a correction factor that would allow a direct comparison between suppliers' activity levels. So another group of products that Akron has been expanding over the years is the human blood-derived products as supplement medias. And as human-derived, these supplements present another challenge, which is safety. Especially these days that we are living in with a global pandemic, it is crucial as a provider to ensure a reliable supply of safe product. A result of this commitment is the recent launch of our newest human AV serum, jointly developed under the exclusive agreement with Octapharma. This product starts from Octaplus, an FDA-approved, sterile, pyrogen-free, solvent detergent treated pooled plasma from US donors. It is compliant with sterility tests per United States and European pharmacopoeias. It is fully xeno-free and manufactured under ISO 13485. And the larger pools that are used in this product offer a very high batch-to-batch consistency. But the main feature that makes this product revolutionary and outstanding is that since the starting material is Octaplus, the resulting AV serum is virus inactivated. Octaplus has a very large donor pool size. Donations are tested per 21 CFR 610.40 for biomarkers using FDA licensed serological tests. Additional screening programs reduces the risk further. The solvent detergent method inactivates enveloped viruses. The larger pools that I was mentioning achieves robust levels of neutralizing antibodies for immune neutralization. And the affinity chromatography, together with multiple sterile filtration steps, remove potential pathogens. So it is the compilation of many steps along the manufacturing process that achieves the required viral inactivation of both enveloped and non-enveloped viruses, conferring this required safety and reliability. So again, Akron wants to demonstrate the high quality of its products in terms of safety, but also performance. And this is a study that shows that the new viral-activated AV serum has an equal, if not superior, performance when compared to some available alternatives in the market. In summary, Akron, together with Octapharma, has developed a revolutionary product that is not only safe and compliant with the regulations, but also with a great performance and backed by a highly robust supply chain. Another supplement media that has been optimized for commercial manufacturing and that puts Akron in the front line is the human serum albumin. This product is derived from the cone fraction 5. The donors have been tested per approved guidelines and collected from FDA licensed donor centers. Since it does not contain stabilizers like sodium caprylate or sodium N-acetyl tryptophanate, it has been optimized for cell therapy further manufacturing. It is GMP manufactured in an ISO-certified facility, and it is supported by an electronic DMF filed with the FDA as well. Unlike the cytokines, Akron is also offering the HSA in weldable bags to suit functionally closed systems, providing a very versatile packaging. And we will actually have the first batch of HSA in bags in Q2 2021. So at Akron, we know that in the cell and gene therapy field, there is no one-fits-all solution. So we also offer custom services to help on the development of exclusive pieces that will contribute to distinctive therapies. And we want to accompany all sizes of companies, from the biggest ones to academic groups that are starting their journey towards an IND filing. We have many years of expertise on the customized manufacture of recombinant proteins, medias, transfixion agents, human plasma-derived products, and DNA plasmids or nucleases. And we are actually currently developing some Cas-related nucleases. As I mentioned, in 2020, we have built a new facility focused on the plasmid manufacturing, offering 3 different grades of plasmids, from research to fully GMP, that will be able to cover commercial scales. The facility is 60,000 square feet. 15,000 of these are used in clean room spaces, and it is equipped with quality control and process development labs, having plenty of space for expansion. So the facility comes with top-notch technology thanks to the collaboration with Cytiva, having very robust upstream processes with single-use manufacturing platforms, and multiple chromatography steps on the downstream processes that ensure a highly pure product. So we will not only be able to produce research-grade plasmids, but also high-quality and GMP-grade, which will count with GMP controls with segregation and traceability, 21 CFR Part 11 compliant QMS, validated QA oversight and TSC BSC statement, GMP compliant production suites, single-use platform, and certificate of analysis and manufacturing summary report. And the main difference between high quality and GMP grade is that the latter will go through the whole process, development process, with all the engineering runs, and it will get a certificate of analysis and project-specific master batch record. So, in summary, Akron continues to optimize ancillary materials to address key bottlenecks and support the industrialization of cell and gene therapy production. And how we do that? Developing functionally closed systems at desirable fill volumes and concentrations, prioritizing safety and consistency with virus inactivated human blood-derived products, increasing transparency and regulatory support with electronic format drug master files, and addressing key industry bottlenecks such as GMP plasmid DNA manufacturing. And with that, thank you all very much for your attention. Any question, please feel free to contact me during the conference or through this email address, and you are more than welcome to pass by our booth. So thank you very much.
So hi, Sara, I have one question. Are any of your products meant to be used as an excipient? For example, your albumin product, I know it's mentioned on your slides for further manufacturing. I think a lot of us out in the field are looking for albumin that we can also use, as excipients for delivery or in cryomedia. Do you have thoughts on that?
So yeah, for example, we have in the pipeline to have, for example, the IL-12. We are bringing it as a therapeutical grade. All the reasons that we have at this point, they are intended for using further manufacturing for cell culture. Nonetheless, for example, we are currently developing cryopreservation medias that will be able to be infused in the patients. Yeah.
Okay, great. Thank you. Are there any other questions for Sara? Okay, great. Well, thank you, Sara. That was a great presentation.
Thank you very much.